Hello everyone and welcome back. I am with the second part of this video. Hope you understand the first video where we really laid down that biological foundation. Today we're taking things a step further. I want you to imagine the human digestive system as a highly specialized, incredibly efficient dismantling factory. You see, your body's primary job after a meal is to break down massive, complex structures into tiny, usable pieces of energy. But what happens when the machinery in this factory suddenly malfunctions? Well, let's dive into this fascinating microscopic world and find out.
Here's our roadmap for today's explainer. We'll start with the carbohydrate journey and absorbing the simple sugars. Then we'll look at what goes wrong in the pathology of malabsorption, followed by specific deep dives into lactose intolerance and sucrase isomaltase deficiency. Finally, we'll wrap up with diagnosis and dietary treatments.
All right, let's jump right into section 1, the carbohydrate journey.
To truly grasp this topic, we have to start by asking a pretty fundamental question. What actually happens when our body's sugar-digesting machinery breaks down? Carbohydrates account for about 40 to 60% of the calories in a typical Western diet, but because they're so complex and hydrophilic, they require a very specific, perfectly timed sequence of reactions to become useful fuel. If that machinery fails, the consequences are immediate and honestly incredibly uncomfortable. Let's look at the very first step of this dismantling process. I want you to think of enzymes as biological molecular scissors. The second food enters your mouth, salivary amylase starts snipping away. Then pancreatic amylase takes over once things reach the small intestine. These enzymes target complex carbohydrates, like starch, and literally chop those long chains into smaller, more manageable blocks, such as limit dextrins and a disaccharide called maltose. But here's the catch. These pieces are still way too big for your body to actually absorb. So the food moves along to the microvillus membrane, which is commonly called the intestinal brush border. Here we have these highly specific enzymes, like Maltase, Sucrase, and Lactase, just waiting to make the final cuts. They take disaccharides, which are basically double sugars, and snip them down into simple, single monosaccharides like glucose and fructose. Our bodies can only absorb monosaccharides. So without these exact microscopic scissors working right at the brush border, all that fuel is completely useless to us.
Moving on to section 2, absorbing the simple sugars.
Once our molecular scissors have done their job, we need to get these newly minted simple sugars out of the gut and into the bloodstream. And they face an incredibly complex cellular membrane to get there. They essentially need specialized doors to get inside the enterocyte, or the intestinal cell. For glucose and galactose, that door is a transporter called SGLT1, which actively pulls the sugar inside right alongside sodium. Fructose uses a totally different door called GLUT-5. And once they're inside the cell, they all exit into the bloodstream via yet another transporter called GLUT-2. It is a highly orchestrated event. So just to quickly recap this normal, healthy physiological journey. Step 1, we ingest carbohydrates and salivary breakdown begins. Step 2, enzymatic hydrolysis makes the final cuts at the intestinal brush border. Step 3, simple sugars are transported into the enterocyte through SGLT1 or GLUT-5. And step 4, they're released into the bloodstream via GLUT-2 for energy. That right there is the healthy assembly line functioning perfectly. All right, let's get into section 3, the pathology of malabsorption. This is where things get messy. What physically happens when those scissors are missing or the doors get jammed? Well, when an enzyme is deficient, the undigested sugars just sit there in your intestinal lumen. And because they're osmotic agents, they start drawing excess fluid directly into the gut. Then, these sugars travel down into the colon, where billions of bacteria are waiting for a free meal. The bacteria aggressively ferment these unabsorbed sugars, releasing massive amounts of gases, like hydrogen and methane, along with short-chain fatty acids. This biological fermentation cascade is exactly what causes the severe bloating, flatulence, cramping, and osmotic diarrhea that patients experience. Which leads us to section 4, lactose intolerance explained. To really understand this process, we have to look at the most common enzyme deficiency in the world. Lactose is a disaccharide found in milk and dairy. In a totally healthy gut, the lactase enzyme acts as the scissor, splitting lactose into galactose and glucose. But if a person has a lactase deficiency, this vital process simply stops. The lactose is untouched, it can't be absorbed, and it immediately triggers that painful fermentation and fluid drawing cascade we just talked about. We generally categorize this breakdown into 2 types. Primary lactase deficiency is essentially a genetically programmed age-related decline. For most mammals and a lot of human populations, lactase naturally drops off after childhood, heavily influenced by genetics. On the flip side, secondary lactase deficiency is acquired. If you get a severe stomach bug, take certain antibiotics, or have celiac disease, the intestinal lining, where these enzymes actually live, gets injured. The really good news here, once the underlying injury heals, secondary deficiency is often completely reversible.
Let's shift gears a bit to Section 5, Sucrase Isomaltase Deficiency.
So while lactose intolerance is incredibly common, genetic sucrase isomaltase deficiency, or GSID, is a lesser known but increasingly recognized condition. It's an inherited disorder where the body simply lacks the specific enzyme machinery to break down sucrose, which is your standard table sugar, and sometimes starch. Historically, doctors thought this was a very rare condition that only affected infants. But growing evidence is showing us that genetic variants of this enzyme are a huge emerging piece of the puzzle for a lot of people dealing with unexplained gut issues.
If you look closely at the structure of the sucrase isomaltase protein on a cellular level, you'll see that pathogenic mutations can occur all over the place. Sometimes the protein gets stuck inside the cell and never makes it to the brush border. Other times it makes it there but just doesn't work right. These genetic defects quite literally break the molecular scissors, leaving the patient totally unable to process standard dietary sugars.
Because there are so many different mutations, the amount of working enzyme left over can vary wildly from person to person. And that means the clinical presentation changes significantly as a patient ages. Infants who have shorter intestines and higher metabolic needs might show severe diarrhea and failure to thrive as soon as they are given solid foods. But as patients grow, the symptoms evolve. Adolescents and adults might just experience excessive gas, bloating, and symptoms that perfectly mimic diarrhea-predominant IBS.
Finally, let's wrap things up with Section 6, Diagnosis and Dietary Treatments.
If a doctor suspects the factory machinery is broken, the gold standard for diagnosing it is taking a biopsy of the intestinal tissue and directly measuring the enzyme activity. But there's also a great non-invasive alternative, the hydrogen breath test. Remember those fermenting bacteria producing gas? Well, that hydrogen enters your blood and is exhaled right through your lungs, meaning doctors can literally measure the unabsorbed fermenting sugars on your breath. Once diagnosed, the first-line treatment is simple but definitely tough: strict dietary avoidance of the offending sugar. Luckily, we also have enzyme replacement therapies.
When it comes down to treatment, the approach really depends on which enzyme is missing. For lactose intolerance, patients use lactose-free products or take a supplemental lactase pill right before eating dairy. For GSID, patients restrict sucrose and initially starch, but they can be prescribed an enzyme replacement called Sucrase. It's derived from baker's yeast and replaces the missing sucrase activity, though they still have to monitor starch intake. In both scenarios, targeted medical therapies are literally stepping in to replace the specific missing molecular scissors.
We have covered a ton of ground today, from the healthy, synchronized dance of carbohydrate absorption to the painful cascade of malabsorption. So I want to leave you with this final thought. Is your gut trying to tell you something about your own microscopic machinery? The next time you experience an upset stomach after a meal, you're going to know exactly what's happening on a cellular level between your enzymes, your transporters, and your microbiome. Thank you so much for exploring the factory with me today, and keep learning.